Okay, this is the biochemistry quiz and test review video. I will be letting you know when we get to the end of the material that will be covered on the quiz, and then when we start talking about the new topics that are on the unit test but aren't on the quiz. Students should be aware that at any time on any unit test, we might ask experimental design questions that require you to be able to identify the hypothesis, the independent and dependent variable, and also the control group. So what is an organic versus an inorganic compound? There are two elements that are always found in any organic molecule, and those two elements are, can you come up with them in your head before I give this? Carbon and hydrogen. If it doesn't have both carbon and hydrogen in it, it cannot be organic. So what are a few other elements that are often found? You're often going to find oxygen, nitrogen, phosphorus, and sulfur. But there must be carbon and hydrogen. So where is energy stored in an organic molecule? In the bonds between the carbon atoms and in the bonds between the carbons and the hydrogens. Now, it's not quite as simple as that. You'd need a lot more chemistry to really understand how this works, but the ability to break those carbon to carbon bonds and form new bonds, or break those carbon to hydrogen bonds and form new bonds, is really how chemical energy is stored. So if you are given a list of compounds, you ought to be able to determine which are organic and which are inorganic. One thing that often throws students for a loop is that they think water is organic because they think if it is in their body and in living things in large quantities, it must be organic. They think of organic as living things, but water has hydrogen and oxygen in it, right? It has hydrogen and oxygen, but water has no carbon in it. So water is actually an inorganic compound. And in addition to the fact that water doesn't have carbon, water is fairly simple, and most organic compounds are more complex in terms of the number of atoms. So what type of chemical reaction is required for the synthesis of polymers and also actually dimers? That would be dehydration synthesis. If you have smaller molecules coming together to form larger molecules and water being released, you've got dehydration synthesis. Dehydration means removing water, and synthesis means putting together. So one of the things that throws students off is they use the answer dehydration synthesis early on the test and quiz and then think, oh, I already used that answer, it can't come into play again. But in fact, anytime you have the production of dimers or polymers, you're having dehydration synthesis going on in this unit. Okay, so what type of chemical reaction is responsible for the breakdown of dimers and polymers into monomers? That would be hydrolysis. Hydro means water, lysis means destruction. So it's using water to break bonds. And the hydrogen and the H and the OH actually are added to those molecules where the bond breaks. So you need to be able to distinguish between diagrams of these two types of reactions. So if water is going in, you have hydrolysis. If water is coming out, you have dehydration synthesis. If two smaller molecules are coming together to make a bigger molecule, you have synthesis. If a bigger molecule is being broken down into smaller molecules, you have hydrolysis. So this is an animation of dehydration synthesis. The hydrogen and hydroxide, so the H and the OH, come off to make room for the bond to form between the two molecules. So this would be a polysaccharide. If we go back to the beginning, we go from two monosaccharides to a disaccharide, and now we would have a polysaccharide. Remember that the prefix mono means one, di means two, and poly means many. So this is an animation of the reverse. So water comes in, and the hydrogen and hydroxyl group, the H and the OH, actually replace the bonds that were formed between the two molecules. So this is hydrolysis. So let's talk about water for a little bit. So what type of bonds hold the hydrogen atoms to the oxygen atoms in a single molecule of water? So within the water molecule, that would be a polar covalent bond. And remember, those are the bonds where you've got unequal sharing of electrons. So what type of bond holds the hydrogen atoms of one water molecule to the oxygen atom of another molecule of water? So the attraction between the oxygen and the hydrogen, that's referred to as a hydrogen bond. So that one is a polar covalent bond, and that one is the hydrogen bond. Chemistry teachers will be quick to tell you that this isn't really technically a bond, it's really an attraction. So just remember that when you get to chemistry next year. And by the way, this diagram is also showing the cohesion of water. When you have two water molecules sticking together, that's cohesion. If you have a water molecule sticking to a xylem cell, or anything else, that's adhesion. So what are the building blocks of lipids? Lipids, and they're also called triglycerides or fats, are built from one glycerol and 3 fatty acid molecules. So this shows you a diagram of a triglyceride. You can see the 3 fatty acids and the glycerol molecule. So that's our glycerol, and those are our 3 fatty acids. Be really careful because sometimes students confuse glycerol with glycogen. This is not glycogen, it's glycerol. Be really careful. There are a lot of words that start with GL. There's glucose and glycogen and glycerol, so just be careful to be able to distinguish between all of those words. So now I'm going to show you the formation of a triglyceride by dehydration synthesis. So we have water forming. In order to make room, those hydrogens and hydroxides come off to make room for the new atoms to bond. So this is a saturated fatty acid, this is a saturated fatty acid, and this is an unsaturated fatty acid because of this double bond here. So molecules containing which type of fatty acid are solid at room temperature? That would be our saturated fatty acids. And here's a diagram of a saturated fatty acid. Notice all of the carbon-to-carbon bonds are single bonds. And there's lots of hydrogens in this molecule. Beware that the carboxyl group, this COOH, does have a double bond between the carbon and the oxygen, but that doesn't count. It's not a carbon-to-carbon bond. It's a carbon-to-oxygen bond. So there are no double or triple bonds between any of the carbons in this chain. You can also tell because the chain is straight and isn't bent. Molecules containing which type of fatty acid would be liquid at room temperature? So that would be our unsaturated fatty acids. Now be careful because this is not an unsaturated fat, it's an unsaturated fatty acid, which is a component of an unsaturated fat. And so this would be found in the triglycerides in oil, like olive oil. This would be found in the triglycerides in animal fats, like butter or lard. So molecules containing which type of fatty acid are found mostly in plants? That would be our unsaturated fatty acids. So this is like our olive oil and sometimes found also in avocado. So molecules containing which type of fatty acid are mostly found in animals? That'd be our saturated fatty acids. Which are healthier? That'd be our unsaturated fatty acids. Now, this is somewhat controversial, and there's been some discussion back and forth about, which types of fats are healthy. And obviously, the omega-3s are really good. Those found in fish oils, but oftentimes the fats found in like steak and especially in processed meats are really bad for you. And extra virgin olive oil is pretty good for you, but it seems like the more we tamper with a fat in the manufacturing process, the worse it ends up becoming for us. So molecules containing which type of fatty acid are more unhealthy? Saturated fatty acids. And they're found in lard and butter, so animal products like cheese and heavy whipping cream. Molecules containing which type of fatty acid are found in olive and canola oil? That'd be our unsaturated fatty acids. So it's unsaturated because there is a double bond between these two carbons. And this one is a mono-unsaturated because there's only one double bond. And when we say unsaturated here, we're talking about it's unsaturated with hydrogens. If you broke this double bond, you could add 2 more hydrogens because remember, every carbon can form 4 bonds at once. So if you're talking about a sponge being unsaturated, it could hold more water. And in this case, if a fatty acid is unsaturated, it could hold more hydrogens, but only if you broke the double bond. And remember, we still have the double bond between the carbon and the oxygen in this carboxyl group, but that doesn't count in terms of our saturation. So what type of molecule comprises most of the cell membranes in all cells? This one is super important. This would be a phospholipid. So you can tell it's a phospholipid because it has this PO4, this phosphate group right here. So always look for that. That's the telltale sign that you probably have a phospholipid. It doesn't guarantee that, but you're looking for a glycerol chain right here, this 3-carbon chain, a phosphate group, and 2 fatty acid tails. Remember, triglycerides have 3 fatty acid tails. So not all fats have 3 fatty acids and glycerol. That's just the most common type of fat. So again, we have our phosphate group. Our glycerol chain is right here. And we have our 2 fatty acid tails. This one is saturated and this one is unsaturated. And these make up, if you have a lot of them, the phospholipid bilayer that makes up the boundary of cell membranes and separates the inside contents of a cell with the external environment. So you ought to be able to visually identify a phospholipid. Be careful because sometimes the diagrams will be sideways or upside down. Remember that these molecules are 3-dimensional and rotate in space. So don't expect to see the exact same diagrams from your notes on the test. Sometimes we find alternative diagrams. You ought to be able to recognize any phospholipid based on its components, even if it's drawn differently by a different graphic artist. So you're going to be looking for your 2 fatty acid tails, your glycerol chain, and your phosphate group. So here are 2 other diagrams. We want you to be able to open up any biology book and recognize these molecules, even if the diagram is a little bit different. So here we have our phosphate group and our 2 fatty acid tails, and here is our glycerol. It's actually bent. And in here it's drawn straight. This is more accurate in terms of the shape of the molecule. Now, one of the things you'll notice is that this molecule is showing you all of the carbons and hydrogens. This molecule is really the way an organic chemist might draw this for other organic chemists, because they know that there are hydrogens here, and they know that everywhere you have a bend here that there would be a carbon, right? But they're not showing you all the hydrogens, and oftentimes organic chemists and biochemists don't take the time to draw every hydrogen because we all understand they're there, we know they're there, and so it's kind of like a shorthand. So here's our glycerol backbone, our phosphate head, and our fatty acid tails. So now we're going to shift into our next topic, carbohydrates. So the general term for the monomer of carbohydrates is a monosaccharide. That is a general term. So the family that we're talking about is carbohydrates. Now we're going to talk about some branches of the family. So we have our monosaccharides. Mono means 1. Saccharide means sugar, so it's our simple sugars. This is glucose, and that is fructose. We don't typically require you to be able to tell the difference between glucose and fructose on the test, but we do usually make sure that you know that if you look at either of those, that they're monosaccharides, and you can tell because they're 1 ring. Now, glucose is a hexagon shape, and fructose is a pentagon shape, so students often do not have a hard time distinguishing between those two, but we usually don't test on that. We could ask you, is glucose a monosaccharide or is fructose a monosaccharide, and you need to know that definitely. So glucose and fructose are both monosaccharides. They're also monomers. A monomer is just a general term for any building block or single unit of something. So monomer is very general. Monosaccharide is a little bit more specific because it tells you we're talking about carbohydrates. And then glucose and fructose are very specific. Any organic chemist knows exactly what they are based on their name and could draw them probably from memory. So the general term for the dimer of carbohydrates would be disaccharide, and that is sucrose, and that one is maltose. So a sucrose is a glucose bonded to a fructose, and maltose is two glucoses bonded to each other Lactose, the sugar found in milk, is an additional disaccharide, which we're not going to test you on, but students often kind of wonder, well, what about lactose? Because they've heard of lactose, it's in milk, and they've heard that some people are lactose intolerant. One thing that you might notice is that carbohydrates tend to end in ose at the ending. Okay, so like maltose, glucose, sucrose, fructose, galactose. And you can tell that it's a sucrose because it has a hexagon bonded to a pentagon, whereas maltose is two glucose, so it's two hexagons bonded together. Now, these would have been formed by dehydration synthesis that would form this bond. So any dimer or polymer is formed by dehydration synthesis. The general term for a polymer of carbohydrates is a polysaccharide. Remember, the prefix poly means many. That is our friend cellulose, and that is starch or glycogen. So if you look at the cellulose, you'll notice it has no branches, and that's one clue. But I could just be showing you a small piece of a starch and, like that, and you wouldn't necessarily know. So that's not what you're going to look to as the telltale. Like if it's branched, you know it's not cellulose. But if it's not branched, you can't be sure it's cellulose. What you really want to be looking for is if you look at the glucose molecules, they alternate in directions. So the oxygen is in the top on this one, and then the bottom here, top here, bottom here, top here. So they alternate directions, right side up and upside down. In starch and glycogen, if you look at the oxygens, they're all facing the same direction. And that changes the bond angles between them. And enzymes in the human body and in most organisms are really good at breaking this shape bond, because shape determines everything in it with enzymes, but not so much the cellulose. So cellulose is undigestible by most organisms. The important thing about cellulose in nature is that it is the cell wall component in plants and algae. And sometimes students get phospholipids and cellulose confused because phospholipids are the building blocks of cell membranes, and this cellulose is the building block of cell walls. So there's a phospholipid bilayer, and if it's a plant cell, outside that phospholipid bilayer, surrounding that phospholipid bilayer is an additional protective layer of cellulose that gives the plant cell or algae cell, a sense of really rigid structure. So I like to think cellulose, and then it reminds me of the word celery. Celery is a plant, and it's very crunchy, and that's kind of how I think about cellulose. So starch and glycogen's job is for energy storage. So plants will store starch in their roots typically, like think about potatoes, and sometimes in their seeds, like if you're thinking about cornstarch. And if you think of cornstarch, which you might have in your cabinet for thickening sauces, or if you're making pies, then you can think, oh, corn, that's a plant, and it has, starch, so cornstarch, and that's a good memory trick. Glycogen and starch are very, 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 very similar. They're so similar that in pretty much every textbook, they use one diagram and say, oh, this is starch or glycogen. So the only difference between starch and glycogen is that glycogen has a little bit more of a branching pattern. And that's because if you think about it, animals move, and so we need more energy. And it is actually the glucose that we use to make ATP for energy to do work in our cells. So we basically bond them in long branched chains and then later break them down into individual glucoses that we can break down even further. We cannot break cellulose down, which is why eating lettuce doesn't have many calories. We can't get the energy out of the cellulose in plant cell walls. So plants use starch, animals use glycogen. They're very, very similar, and those are energy storage. Now, they're long-term energy storage to an extent, but even longer-term energy storage would be those triglycerides I showed you earlier, those fats. So we cannot digest cellulose, and so it's considered dietary fiber. It is really good for us to eat things high in dietary fiber for a couple reasons. It, first of all, makes us feel full faster, and we don't have to worry about starvation because we are not hunter-gatherers anymore, and our hunting and gathering is basically going to the grocery store. So cellulose makes us feel full, and then we don't end up overeating and getting a lot of sugar and starch in our diet. because it makes us feel full longer. Also, if you eat something that is starchy and has cellulose, having the cellulose in there makes you digest and absorb the sugar at a more slow, steady pace, which is good. Cellulose is also good for preventing colon cancer because it helps keep you regular in terms of eliminating your solid wastes, in other words, pooping. But in addition, most foods that are high in cellulose are also high in vitamins and minerals and other nutrients that you need, like plants, vegetables and fruits. So we want to be eating things with lots of cellulose, less with our starch. So cellulose, again, is cell wall structure in plants and algae. So now I want to talk about proteins. So proteins have the monomer called amino acid. Be really careful because sometimes students hear nucleic acids and amino acids and think that they're related because they are both acids. But in fact, lots of these things, anything that I've shown you that has a carboxyl group is also an organic acid. And so there are lots of these biochemical compounds that are organic acids. So amino acids are called that because of this NH2 here. This is the amine group. This is our central carbon. There's always a central carbon and a hydrogen, an amino group and a carboxyl group. And then we have this variable group referred to as the R group. And there are 20 different variations, the simplest being just a hydrogen, and the more complex, there's actually a ring in phenylalanine. So the process that bonds two monomers into a dimer would be, again, dehydration synthesis. That's a common theme you'll see throughout this entire unit. So I'm going to show you dehydration synthesis between these two molecules. These are two of the simplest amino acids because the variable group is just a hydrogen. And the H and the OH come off to make room for the bond between the carbon and the nitrogen. You can always tell where the bond is by if you find the carbon that is still double bonded to an oxygen and also bonded to a nitrogen, that is your peptide bond. And you also can tell how many amino acids you have by counting the number of nitrogens. So there's 2 nitrogens, so this is a dipeptide. So it's called a dipeptide if you have 2 amino acids bonded together. Again, you can count the nitrogens, 2 nitrogens, prefix di. The name of the bond is called a peptide bond, so that's the bond that is showing here. And the polymer of this molecule is called a polypeptide. And again, we're not showing you all the details of it, but each of these little circles can be considered, or it represents an amino acid. So polypeptides are not considered proteins until they fold into their 3-dimensional shape. So it goes from amino acid to dipeptide, dipeptide to polypeptide, and then it folds into its 3-dimensional shape, and then it is a protein. So I am going to switch, without pausing, I'm going to switch PowerPoints and open a different PowerPoint because I'm going to review some structures with you guys. I'm going to go back to the top. So, and I'm going back to the structures that you need to know for the quiz, also the test, but I'm going to first review for the quiz and then add the stuff that's on the test to the second half of the review so that you can stop reviewing if you're reviewing for the quiz without looking at the topics you haven't been taught yet. So this molecule would be water. The bond that holds the hydrogen and oxygen together within a water molecule is called a polar covalent bond. The bond that, or the attraction between the negatively charged oxygen of one water molecule and the positively charged hydrogen of another water molecule is called a hydrogen bond. You can always tell hydrogen bonds in diagrams because there is a dotted line or a dashed line. In this case, you have a dotted line, but sometimes it's a dashed line. These 2 molecules would be carbohydrates, to be most general. They are monomers, that's still pretty general. They are monosaccharides, that's more specific. And if we want to get very specific, we could say that's glucose and that's fructose. So to what class of organic compounds do they belong? They are monomers of carbohydrates. Pretty much though, All of the complex carbohydrates that we have studied are long repeating chains of glucose. That's a common theme, that glucose is stuck together in a variety of different shapes. So that's glucose and that's fructose. Starch, glycogen, and cellulose, those are all polymers of Mr. Glucose over here. So what kinds of molecules are these? Well, we know they're dimers because there's 2 rings. We know they're disaccharides because there is a 2-to-1 ratio of carbon or hydrogen to oxygen. So if you hear the term carbohydrate, it tells you it's carbon with the same ratio of hydrogen to oxygen as water. Hence, if you add water to something, you're hydrating it. One of the things that I just wanted to point out is they're not showing you all the carbons. Everywhere here in the corners where you have a point in these hexagons or pentagons, there's a carbon that they're not showing you. All organic chemists or biochemists or students even know that there's carbons there, that they're just not all drawn. So notice that our disaccharides, you can tell because they're ring structures and they have 2 rings. How are disaccharides formed? The same way every dimer or polymer is formed. Anytime you're putting together 2 individual monomers to make a dimer, it's going to be dehydration synthesis. So in this case, it's dehydration synthesis between 2 monosaccharides to make a disaccharide. That is maltose, that's its specific name. So disaccharide just is a category, but maltose is the specific disaccharide when you have 2 glucoses bonded together. And sucrose is the disaccharide when you have a glucose and a fructose bonded together. I have always thought that this kind of looks like an ant. So disaccharides are carbohydrates we're still in the same family here. So these would be polysaccharides because they have many rings. This one on the top could be one of 2 things. It is definitely a complex carbohydrate because it has many rings, but this would be starch or glycogen. Animals store energy in the form of glycogen. Plants store energy in the form of starch, and the reason I have used green here is because plants photosynthesize, so they have chlorophyll, so they're green. Now this down here is cellulose, and it is the major structural component of cell walls in both plants and algae. And again, you can tell it is cellulose because it has the oxygens in alternating directions, and so it's going to have a different bond angle because each of these glucose alternate directions, so right side up and upside down. These, if you notice, all of the oxygens are facing the same direction. You need to be able to distinguish between all of these structures on tests and quizzes. We can't digest cellulose, so it's considered dietary fiber. And remember, we learned about the mutualistic relationship earlier when we had our microorganisms that were living in the digestive tract of the termites. So remember, the termites can't digest cellulose. The single-celled protozoans that live in their gut can. And so that's the mutualistic relationship because the cellulose is broken down into glucose so that the insect can actually get some energy from it, but then the insects are bringing a constant supply of energy to those protozoans by eating the wood. So the rings alternate direction and there are no branches in cellulose. So the reaction at the top, that would be dehydration synthesis. How can you tell? You have 2 monomers, right? And the H and the OH come off forming water. So water is coming out and you have 2 monomers and you're making a dimer. The one on the bottom would be hydrolysis because you have water going in, breaking a bond, and you're starting off with a disaccharide, sucrose specifically, and it is breaking it into glucose and fructose. So it is a monosaccharide, or disaccharide broken down into 2 monosaccharides. So you have a larger molecule being broken down into 2 smaller molecules by adding the hydrogen and hydroxide groups from water, so the HOH. What kind of molecule is that? That would be a lipid, also called a triglyceride because you have tri is for the 3 fatty acids and glyceride for glycerol. It is also formed by dehydration synthesis between glycerol and 3 fatty acids. So here's our glycerol. Here's our 3 fatty acids. Fatty acids can be saturated or unsaturated. This one is a saturated fatty acid because all of the carbon-to-carbon bonds are single. right? And it's full of hydrogen. No more hydrogen can be added. We still have this double bond here in the carboxyl group, but that doesn't count because it's not between carbon, it's between carbon and oxygen. So all the bonds between carbon atoms are single bonds, and triglycerides made with saturated fatty acids are solid at room temperature. So this itself isn't a fat, it's a fatty acid. On the previous page, this is a fat. Fats are made of glycerol and 3 fatty acids. So this is a component of a fat. They are found in animal fats like lard or butter, and they're pretty bad for your health. They're converted by the body into cholesterol, which we're pretty sure causes heart disease. So that's actually one interesting thing. That's why if you are on Lipitor, you take it right before bed at night, because that's when your body would be converting those saturated fats into cholesterol. They used to think dietary cholesterol was really the problem, but it turns out it's really the saturated fat that your body then later converts into cholesterol. So fatty acids can be unsaturated or saturated, which is this. That will be unsaturated because there is a double bond between these 2 carbons. And in fact, more specifically, this is a mono unsaturated fatty acid because you have 1 double bond. So right here, and usually these are drawn bent, although sometimes they won't. Sometimes they'll put one of the hydrogens on the opposite side and make it straight, but usually they're drawn with a bent shape. And triglycerides made with saturated fatty acids are liquids at room temperature. They're found in plant fats like olive oil or canola oil. And they tend to be good for your health in small amounts, particularly the monounsaturated fats found in extra virgin olive oil. But again, we still want to keep this to a low amount in our diet. We need some fat in our diet. We just, as Americans, probably have too much fat in our diet. We don't want to go fat-free, but we want to make sure that we're eating the healthy kinds of fats. So for example, we want to be eating fish, we want to be eating avocados, we want to be eating nuts and extra virgin olive oil, and we want to be eating less butter and cheese and full fat milk, although once in a while you gotta splurge a little bit. We just maybe should eat less of those things in general. So this is a phospholipid, and you can tell because it has a phosphate group, a glycerol, and our 2 fatty acid tails. And the structure of cell membranes is made up of a double layer of these phospholipids. This part is hydrophilic, it means water-loving. It's attracted to water. The hydrophobic tails are repelled by water, and so this leads to 2 layers of lipids where the tails are facing each other, and the surface on the outside and the inside is covered with the hydrophilic water-loving heads. So this diagram is a cross-section of a cell membrane, and so there would be water outside the cell, there would be water inside the cell, and those hydrophobic tails, they're water repellent, flip towards each other, and the heads that are attracted to water flip towards the outside because just they're chemically attracted to water. And these guys, by the way, are proteins. This is a transport protein. We'll get more into that later. And this is a glycoprotein because this is a carbohydrate chain. We're going to get more into how that's used for cell-to-cell communication in upcoming units. So there are 2 layers of phospholipids, hence the phospholipid bilayer. Bi means 2, so there's 2 layers of phospholipids. You can see here's one phospholipid and here's another phospholipid, and the tails are facing each other. This just happens naturally if you put phospholipids in large numbers in some water. So the hydrophilic heads face the inner and outer surface of the cell, and the hydrophobic tails are on the inside of the membrane away from all the water, whether it's inside the cell or outside the cell. This is an amino acid, and it is the monomer of proteins. That's our central carbon atom. What is that? That is our amino group, and that part is our carboxyl group. Now you may have noticed that the carboxyl group is also, if we go back a few slides, found on our fatty acids. So amino acids, fatty acids, the reason they both end in the word acid is because they have this carboxyl group. And acids, actually, the hydrogen comes off, and anything that, if you put it in water, releases hydrogens is an acid. So that's our amino group and our carboxyl group, and that is our variable group. There are 20 different variable groups that make the amino acids different from one another. This is a dipeptide. I can tell because there are 2 nitrogens. And this bond here between this carbon and nitrogen is a peptide bond, and I can always find it in a diagram because it's a carbon double bonded to an oxygen that is also bonded to a nitrogen atom. A water molecule is lost when forming a dipeptide. So what type of reaction is this? It's dehydration synthesis. So this shows the different levels of protein structure. We have the primary structure, which is the amino acid sequence. And then that primary structure, which is the chain of amino acids, and the sequence of amino acids is really important. It will fold into several different types of shapes. This is an alpha helix, and this is a beta pleated sheet. And the amino acids then fold into a 3-dimensional tertiary structure. And generally it's a globular shape. All proteins reach at least the tertiary structure. Now, if you change the primary structure, it affects the secondary and the tertiary structure. Just like if you are doing a math problem and you make a mistake early on, it makes everything after that incorrect. The same thing happens with proteins. If you get one wrong amino acid in here, if you're missing an amino acid, you have an extra amino acid, or if you have a different order of amino acids, that changes the protein's shape and always will change the function. So the quaternary structure is not found in all proteins, but some proteins are comprised of more than one amino acid chain, and then they are considered to have a 4th level of structure. So the 1st, 2nd, 3rd, and 4th level of structure. So what is different from one protein to another? And let me tell you, there are a lot of different types of proteins. It is the sequence of amino acids. So all proteins pretty much have all of the exact same 20 amino acids, although there are some proteins that don't have all 20 of those amino acids. But all proteins have the same amino acid alphabet, that 20 different amino acids. But what makes one protein different from any other protein is the order of the 20 different amino acids and the number of amino acids in the polypeptide chain. And I think the smallest, and I'm not 100% sure about this, but I think insulin is 52 amino acids long, and that's pretty small compared to like hemoglobin, which is a really large protein in humans. So we are now ready to pause and take a break, and I'm going to do a second video for the material that is only on the test but not on the quiz. But everything on this video is in the quiz and the test, so make sure you study this again before the unit test.